Welcome to Science with a Twist, a podcast for curious people who enjoy exploring how science impacts our daily lives. From technology that helps the fight against COVID-19 to solutions that help clean the water we drink, it's all thanks to science. In each episode, members of Thermo Fisher's scientific team talk to experts who are on the cutting edge of redefining how we exist. Let's get started.
Hello, everyone. My name is Karen Nelson, and I'm Chief Scientific Officer at Thermo Fisher Scientific. Today, I would like to welcome you to another episode of Science with a Twist. And what we're planning to do today is talk about COVID-19 variants, as well as talk about variants in viruses in general. I'm happy to be joined by two experts who are also my colleagues at Thermo Fisher, Kamini Varma, the Vice President of Research and Development, and Andy Felton, Vice President, Platforms Research and Applied. Kamini, Andy, thank you for joining me today.
Karen, it's great to be here. Thank you for having us over and looking forward to the conversation.
Same. We're very excited to have this conversation. It's a really important topic, so looking forward to having you guys ask us questions and learning more.
And Kamini and Andy, it's been so great working with you since I joined the team. But to give our audience some background on what you do, could you both give us a quick overview of your roles at Thermo Fisher?
So I lead the R&D for our Genetic Testing Solutions Group, which is really focused on infectious disease testing. And as you can imagine, the last few years, we've pretty much centered around COVID testing, whether that's for symptomatic people or asymptomatic, like back to school, back to life type of testing.
And I lead the clinical next generation sequencing division portfolio. And that's the platform, the software capabilities, and all the applications outside of oncology. So next gen sequencing has been around inside Thermo Fisher for about 10 years. I've been with that division pretty much the whole time. But we focus a lot on oncology, but we also cover infectious disease, inherited disease, and a variety of other application spaces as well.
Thank you both. And I know that both of you are really, really deep into the COVID experience and have been critical to the success of everything we've done since the beginning. And, I would like to focus a little bit on the discussion topic of COVID variants, because we hear a lot about different variants, and we all heard a lot about Omicron and Delta. But we've also seen other viruses over our lifetimes that had variants before. So, for example, influenza. And so could you walk us through what it really means when we start talking about variants, and especially, connect to some of the examples we've had before COVID and leading up to COVID?
Yeah, sure. So influenza is obviously an interesting and relevant use case, and particularly because we've had multiple outbreaks of influenza over many years. So many of you may have heard of the great 1918 influenza outbreak. That was a very severe outbreak in the early part of the 20th century that had multiple waves, very similar, in fact, to COVID. Many people are also aware that there are vaccines available for flu, and they change on a yearly basis. And they change because the strains of flu that are circulating in the population change on a yearly basis. And that's because the virus causes influenza evolves. And by that, we mean that the actual DNA or the RNA sequence of the virus actually changes over time due to its evolutionary processes. And what we need to capture in these surveillance efforts is how these viruses are changing and how quickly they are changing. So you've heard of mutations, and the mutations are the result of these changes in the viruses. They change the nucleic acid or the DNA sequence of the virus itself, and that changes the infectivity or the severity of the disease itself. So we monitor these viruses to see how the DNA sequences are changing and whether these changes pose any more threat to the population in terms of infectivity or the severity of disease.
So Kamini, I'm going to turn over to you if you want to add on to that, especially within the realm of SARS-CoV-2.
Yeah, I was gonna add to Andy's comments, right? We're very familiar with influenza. coronaviruses have been around for a while, and we've had like a few of those outbreaks, right, between the SARS and the MERS, but they never really took on the magnitude of a pandemic, unlike COVID. And I think what is unique about COVID is just the level of population spread. It creates this reservoir for the virus to mutate much faster, because initially when COVID came out, we thought it mutated much slower than the influenza virus. But over time, we've seen it pick up speed and momentum, and it's changing much more rapidly. And to the point Andy was making, you could have a change that could be make the virus weaker and it disappears from the population, or you could have a change that makes it stronger and more transmissible. So, for example, with COVID, we've seen, that there's a lot of variants of interest, but the variants of concern are what we've primarily focused on, and those have been the alpha and delta, which was even more transmissible and more dangerous, versus Omicron, I think is far more transmissible than Delta, but has been milder in its infectivity. So I think we've just seen COVID just go through a gamut of changes much faster, and it's brewing in different populations, different geographies, and just spreading very differently in the world.
So, Kamini, could you talk a little bit to the distinction of variants of interest versus variants of concern and how those two levels get separated?
So it has to do with a couple of different factors around health, right? How harmful is it? How fast it's spreading? How transmissible? How quickly it's moving through the population? And really how much of a havoc it creates, or and some of it gets done on a theoretical level first. So, for example, when we saw Omicron and sequenced it, there were so many different mutations in the virus. And knowing the history of viral changes, theoretically, you could predict it was going to be a lot more transmissible. And hence it was given a variant of concern very quickly, because we needed to monitor it and see how it evolved in the population. Thankfully, again, it's not created quite the level of disease as we expected based on our experience with Delta, but that's what differentiates variants of interest from variants of concern.
So I guess a question for both of you is, do we think that variants will continue to arise?
Yes, absolutely. So the virus is evolving, and all viruses evolve constantly all the time, and they're spinning up new variants. Whether those variants rise to what Kamini described, a variant of concern, depends on exactly what changes happen in the DNA sequence. But variants of interest and other variants that have really no effect on what we are concerned about, which is the health outcome of being infected by that organism, happen all the time. So the virus, every time it's mutating inside a person or changing inside a person, is spinning out new variants, some of which may be of interest. And we need to follow them perhaps a little more closely, and some of them may rise to the level of concern in the categorization. And those are the ones that we really think are going to change either the infectivity or the severity of the disease outcome.
So, Kamini, do you have your eyes out on one right now that we might be popping up in the population in the near term?
We're looking at BA2 variant, which is a sublineage of the Omicron. So the lineage that Omicron has 3 lineages, and BA1, BA2, and BA3. And BA1 is the one that overtook the population as a whole. But we started seeing the emergence of BA2, and in places like Denmark and India, it's the primary variant right now. Seems to be a little slower to take over in the US, which has been an interesting thing, because and it's something that we have a lot of speculation around as to why it's not taking over as rapidly in the US as it did in some other parts of the world. And it has to do with the inherent mix of the lineages that exist and which one's stronger, right? So right now we pretty much have our eye on BA2 to see how it evolves.
so fascinating for basic ecologists, right, to see how these viruses can mutate over time. So, Andy, back to you. I'm going to put you on the spot. You mentioned influenza and vaccines on an annual basis. Clearly, this is a different virus. So, what do you think will happen with COVID with respect to vaccination? And if you can't answer, that's fine, but do you have an opinion on that?
Sure, I have an opinion. Obviously, that the scientific community and the CDCs around the world will probably rule on what the eventual outcome is. But it seems like at least we are going into a phase where we're likely to probably have to have annual vaccinations based on the rate that the virus is changing. It's likely that new strains or new variants of concern are going to arise. We know that certain populations who are immunocompromised can generate lots of variants very quickly, and they might get out into the population and cause us to have to rethink the current virus strategies. There's also a possibility that science could really improve very quickly. Vaccine research has gone at an enormous pace, really incredible, since these mRNA vaccines came up. And there is the outside possibility that we can have a coronavirus vaccine that would immunize us against all possible strains in the future. So there's really two outcomes that I see. One is a possible annual shot based on what variants are likely circulating in the population. The other one is the golden ticket, if you will. We'll have one shot that will potentially immunize us against all possible future versions.
That would be nice if that happened.
That would be great if that
right?
could happen.
So I guess the question for both of you, what has been Thermo Fisher's role in the pandemic, specifically with respect to variants?
I could definitely write a chapter on that.
Go for it.
I think, we've been involved right from the beginning, right? So January of 2020, when we heard about the first coronavirus, and mobilized very quickly in China to help the teams there with providing qPCR instruments and working on our first COVID-19 TaqPath assay. And this was when, there were less than 50 sequences in GISAID, and WHO had not even classified it as a virus that we needed to worry about. We already kind of knew that there was something brewing. And we ended up getting our EUA on our TaqPath COVID test in March, and we were one of the first providers to get it. And I feel very proud because we've enabled over 1 billion tests worldwide in the last 2 years, which is phenomenal considering, where and how the teams have had to work in order to scale up to provide this value to the customers. So I think in terms of surveillance, when we designed our assay is unique in that it surveils 3 different gene targets, one of which is the S gene. Our thinking at the time was, hey, this is the most conserved region and something that we want to go after. Well, the vaccine manufacturers had the same idea. So they went after the S gene, and that's been the most susceptible to change. So what we had was a case where when the Alpha variant came out in our TaqPath assay, we saw an S gene dropout. When the Delta variant came on, it came back and converted to a 3 gene assay, and then it again went out. So it kind of became a proxy for looking at new variants unintentionally, but has served us and the community really well in being able to help in surveilling for new variants.
Yeah, I remember when the news came out from South Africa about Omicron, and your test had helped them, as well as sequencing, right, Andy, identify this new variant. Do you want to speak to that? Because, that was, again, one of the key moments in our experience with COVID.
Yeah, sure. Right? So, maybe going back up to what we did in the beginning of the pandemic, similar to Kamini's teams, when there were 40 or so genomes or less in the databases, we realized that this was going to be a severe problem. We needed to design an assay that would surveil the whole viral genome. So we jumped on that very quickly and built an assay called the ANAMPSY SARS-CoV-2 research assay. and made that available to the community to do viral resequencing of the whole genome so we can have a broad picture of what are the changes going on. We've updated that a couple of times. It's now called the SARS Insight Assay. And we've paired that with an easy to use software tool to make sure the community can have access to these genomes very quickly to figure out what's going on and help with the public health effort. To date, we've done over 400,000 genome sequences, which is a phenomenal effort. We have thousands of sites around the world contributing genome sequences. So it's been a big effort and made a big difference in a lot of places around the world. In terms of South Africa, the story is super interesting. Obviously, started with these teams' assays that were seeing some of these dropouts. So we've had a history that one of these particular genes that gets surveilled in the genotyping assays, if there's a change in the viral sequence, that knocks out one component, and that's then a flag, an alarm bell that gets raised that kind of says there's something going on. There's a new strain that's out there that's different than it was before. And then once that information came around, they started sequencing on our Genexus platform. That's a single-day turnaround platform with a very simple, maybe 10 minutes of hands-on time workflow for the lab. And they can immediately see that the Omicron variant was extremely different to those that had seen before. It was very different to Delta. There were an additional 30 mutations in the S gene that were very different than the Delta or the Alpha versions that we'd seen before. And that was the change. And we managed to do that in about 24 hours of them noticing the impact on the tag path detection assay side. So it's a really great story of how the technologies can work together to give us an answer as to what is actually going on in the population.
It's really amazing. And, so the two of you keep bringing up surveillance and data, right? And I want to spend some time thinking that through, because I'm not sure if the networks were up and running at the beginning of COVID. Obviously not, right? And now they have started to come together, and we're hearing about GISAID, et cetera, et cetera. And I know both of you have been critical to the success of integrating data as well as generating data on a massive scale. Andy, you said 400,000 genomes. Yeah. That's unbelievable, right? When we had 17,000 flu genomes a few years ago, we thought that was a great deal. So, but Kamini, I'm going to start with you and, your thoughts on how the community has come together, developed trackers for surveillance of new variants, et cetera. And, just your opinion on what the future looks like. And then if you could talk about some of the partnerships that we're involved in at Thermo Fisher.
Absolutely. It really is a global event, right? COVID-19, and it has brought a very different perspective worldwide. I think we're partnering with a lot of different entities, government institutes. The UK had a very strong surveillance program, as did South Africa, and partnered very closely between both our PCR tools as well as sequencing tools in enabling that effort. Within the US, we have a super strong partnership going on Project ROSA. So this is a partnership with the RADx program at the NIH, with Thermo Fisher providing the different assays and tools in order to detect things. We have 4 different labs in the mix. So University of Washington, Aegis, and some others that can actually run the sequences, and Rosalind, which is helping us with the variant classification and what regions to go after, and actually building out the tracker. So this unique combination of public-private lab partnership has really created the perfect bringing together of solutions as well as the visibility to what is happening in the US. So the Rosalind tracker went live in January, and you could see across the 50 states in the US how the variant is progressing and if there are new things coming on the force. So they're using 2 different methodologies. One is as they run a diagnostic test and get a positive COVID sample, they then take that sample and they can either sequence it or they can run something called a PCR genotyping assay, both of which have different strengths and weaknesses, and together I think gives you a full picture of where and how the variants are going. So I think opportunities like that and collaborations like this are really key in driving insights as well as helping the broader community through better health outcomes. Most of the lay people around now actually understand what an infectious disease is, what testing means, what PCR is, which was not the norm 2 years back, right? And every consumer is getting savvy, so they want to see like what's happening in my community and what's happening in the world around me. And I think we're getting there as far as creating these ecosystems of partnerships. So I'm tremendously excited about it for the future, because we absolutely will have more pandemics, we'll have more outbreaks, and I think next time around we'll be that much better and smarter for it.
And Andy, to follow up on that, I know we have conversations about aerosol scents, and we've seen in the news wastewater testing. Some of your team members are actually involved in these processes. The possibility of doing much more surveillance on a much larger scale, not only for COVID-19, but for other pathogens that could be emergent. So, how do you see us involving in this space, moving beyond some of our current tests and using other applications to look at how the virus is moving around, as well as other pathogens?
Yeah, it's a great question, Karen. So there's a couple of things that you described there that are really super exciting and fascinating and should really help the public health effort in containing the outbreak and trying to understand what's going on. The first of which is wastewater testing. So the sewage systems that are out there in the world collect all our sewage, and it turns out they are a fascinating repository of viruses. And you can actually get the virus out of that material and extract the nucleic acid and sequence it and detect it in those sewage outfalls. And they provide about a 1 week ahead of actual inpatient testing sampling of what is going on in a community. So they're actually a really cool early warning system. And there's a number of, the US is just spinning up a big effort to surveil the sewage systems around the country to kind of give us an early warning outbreak. Europe has been doing this, and some other regions around the world, to really kind of help understand, because as inpatient testing goes down, and maybe it's becoming more endemic, we need a better system to understand what is actually happening and how many people are really getting sick out there. And you can get some idea of that from actually sampling sewage outfall systems. The second one is, what do you do in large congregate settings, where there are lots of people, and you're trying to understand, are we at risk, and is there some action we could take? And that's the aerosol sense program we've involved in. So that is another really cool tool that samples the air, takes liters of air in every few minutes, and it can actually pull the virus onto a filter. And from that filter, we can actually detect its presence and also do sequencing on the material itself and look at what strains are present. So that might help in the kind of large congregate meetings, schools, universities, hospitals. It might help to improve the ability to surveil the presence of those. But on the back end of that, obviously, is the data from all those efforts. And we're also working with a number of groups around the world, including the World Economic Forum, the Rockefeller Institute, who are putting in systems to try and improve the data aggregation so that we get a faster and better view of what is happening in these pandemics. And other test modalities, we can do things like look for multiple viruses, respiratory viruses at one time with either qPCR or NGS-based systems. So there's lots of application that we see coming for these technologies.
I think that's so great to hear, right? So that the knowledge gained from this experience that we all live through together can be, we're not saying there's going to be another pandemic next year, but we just learn from, what works well, what didn't work, how to integrate the data, and what to learn from the data sets, because this is a truly amazing data set that, the nerdy ones in the population would want to learn from. So that's really great to hear that that's happening. And you said that this is also including the developing world, right? So you mentioned that early on.
So the Rockefeller Institute and the WF are looking specifically at low and middle income countries, Africa, Southeast Asia, India, to how we can help, aggregate that data in a way that will benefit the entire continent as well as the rest of the world, right? Because we're not isolated from each other in this pandemic. We're all connected by this virus, and we have to look at the data from everywhere to understand what the future is going to look like.
Yeah. So Kamini, I'm going to close with you. What do you think the future state of testing is going to be as we navigate this at home? Are we all going to get tests at home? Are we going to have combo tests for influenza and flu and COVID at the same time? What are you thinking? And, Andy, chime in if you want, but what do you think the future is going to look like?
I think it'll be a mix of all of it, right? So, we've always, in the last many years, we've moved towards syndromic testing anyway, because we know a lot of viruses present with the same, like, I feel crappy, I have a sniffle, I have, a body ache, right? And, it could be influenza, it could be COVID, it could be just there's like 15, 20, 30 viruses that can result in similar symptoms. So I do think you'll see more of like the COVID flu type tests taking front and center. We have like my kids now, they're like 9 and 10, and they know how to take a swab, and they talk buffer, and they know what's happening, in a tube, and no, mom, let me do it, right? So I think it's the consumerization of these tests is definitely going to be a trend. And, if you could do it easily in the lab, I think, adjusting it to getting more data insights, connecting with public health entities globally, I think there's going to be a trend and shift in all of those areas just because the world is that much smarter.
Yeah. And I think we all got spoiled with doing our doctor check-ins from home by video, right? You realize you didn't have to go into the office every time something was wrong. So I agree with you that it will probably change over time. Andy, you have any thoughts on that?
What Kamini said is really true, right? I think, the consumerization of these tests is going to go very quickly, right? The fact that you can now test yourself at home for one of these infectious diseases means that there are likely to be a lot more of those kinds of tests, and we're going to get a much better picture of what is actually happening in the population around these respiratory viruses or other kinds of viruses and diseases. So I think that can go from viruses to other organisms as well, potentially in the future. So I think we'll see a proliferation of new tests and new capabilities that will help us understand what is happening.
Yeah. So it's clear that viruses are constantly mutating and changing. New variants are inevitable. And because of this, it's important to continue taking protective measures against COVID and help slow the spread. Thank you so much, Kamini and Andy. It was my pleasure to chat with you today. I really appreciate that you shared your insights and expertise on this subject. And if any viewers would like to learn more about Thermo Fisher's response to the pandemic, please visit thermofisher.com. Until next time, this has been Science with a Twist. Stay well.
Thanks for tuning in to this episode of Science with a Twist. This show is brought to you by Thermo Fisher Scientific, the world leader in serving science. If you enjoyed this episode, then follow Science with a Twist wherever you get your favorite podcasts.